All right, we're back with part 2 here of cell organization, function, and so forth in chapter 3. Let's pick up where we left off. In the previous installment of this chapter, we had talked about membrane transport in terms of simple diffusion, facilitated diffusion, active transport, and then a little bit of a variant, facilitated osmosis. I'm going to call facilitated osmosis. It requires protein for it to really be physiologically meaningful. There is a more complex process of moving things in and out of the cell, even in and across the plasma membrane, and it involves secretion and absorption. It's a form of vesicular transport. So a molecule will trigger it's called invagination, where you create a new vesicle that is then internalized. It merges with other vesicles and gets digested and so forth, and you absorb material. There's a bunch going on here. We're not going to get into all of that. If you're moving things into a cell, or sometimes across a cell, or that's called absorption. If you are moving things out of the cell, whether it's back into a digestive tract or it's somewhere else, we call that secretion. It is a form of communication. Like, for instance, you can release things like hormones. For instance, insulin is released in this manner, but it's also a way to obtain and or eliminate materials from the cell. So these vesicular processes do depend on ATP. They are a form of active transport. It requires energy because bending the plasma membrane and pinching it off and merging it is very, very energy intensive. So we have three major processes that we're going to talk about. One is phagocytosis, and there's a subdivision of this that's liquid. They call pinocytosis, but we're just going to stick with phago because that's the only thing in the slide here. This is where you are engulfing, oftentimes it's their objects, like for instance, cell fragments and leftover organelles from, say, a cell got destroyed accidentally, or maybe a pathogen like a bacteria. So neutrophils, if you've ever heard of those cells, they will phagocytose bacteria. Then what happens is that vesicle is then sent to an organelle called a lysosome. And if you have lyso in it, that means break down, just like Lysol, right? Lysol kills bacteria and germs, right? That's where that word comes from. Lyso means break down. So this is basically like the stomach of a cell. You merge your vesicle with the lysosome. Whatever's inside there is going to be subjected to very low pH and or enzymes that'll break stuff down. Very common white blood cells, especially neutrophils, but there are some other ones. So that's phagocytosis. It's internalizing objects. It could be a whole cell or it could be parts of a cell. Then you have endocytosis. This is, these things overlap. I kind of sigh a little bit because phagocytosis is actually a form of endocytosis. This one here, we're endocytosing nutrients. That's true. It's the same thing, only this one is more, shall we say, dead cells and pathogens, but it is a form of endocytosis. Sorry for the confusion. Right? Then we have exocytosis. So this is where we are taking vesicles from inside the cell and sending them to the cell surface for export. Again, all these things require ATP. So again, just a short little 2-3 word phrase should be enough to help remind you what these things are. Okay, that's the goal. Now we have endoplasmic reticulum, which just sounds so abstract as to be somewhat, inaccessible. Here's what it is in a long and a short. Coming off the nucleus and between the nucleus and the plasma membrane, which isn't shown here, there is a reticulum. That is to say, a structure that's kind of got, when you say reticulated, it means it's got all kinds of angles and folds and things along those lines. And so a reticulum, this is a membranous structure. You can see it all around here. You have two flavors of it. You have a rough, which is what the red ones are, and then you have a smooth one. It's endoplasmic as opposed to exo or something along those lines. It's inside the cell. It's a membrane structure inside the cell, different from the Golgi. We'll talk about the Golgi in a little bit. The two major functions of smooth and rough. So smooth is really, again, if you talk about prokaryotes, all of their metabolic processes happen on the inner surface of the plasma membrane. With eukaryotes, you can do that same thing, only you're isolating the enzymes and you're running these metabolic reactions inside the cell rather than on the inner surface. You have a lot more surface area, you can do a lot more stuff. So the smooth endoplasmic reticulum, the key phrase is metabolism. Okay, so you can manufacture lipids in there, you can do steroids, especially in liver cells. Anyway, all right. So there's metabolism, and then there is storage. Okay. When we get to muscle cells, myocardial cells or skeletal muscle cells, they have an endoplasmic reticulum. It's smooth. Only the role of that endoplasmic reticulum is storage of calcium. And that's true in other cells too, but really big time in skeletal muscle and cardiac muscle. All right. What happens is the SER, we call it, will have special channels that will release calcium out into the cytosol in order to turn on and turn off enzymes. So calcium is what we call a cofactor. Either way, though, metabolism, storage, and calcium in particular. All right, that's smooth ER. Rough ER, those red dots that you see on here, not mine, but those ones here in this structure, those are ribosomes. Those are a combination of protein and RNA, ribosomal RNA to be accurate, that are embedded on the endoplasmic reticulum. That's why it's called rough, because if you look at an electron microscope picture of it, those ribosomes look like balls. It does look like some kind of super rough sandpaper. And so you manufacture proteins on the ribosome, which is embedded on the rough ER. You can also make new membranes on the rough ER. That's a little bit of a minor point, but the main thing here, protein synthesis. Once things have been synthesized and or manipulated by the endoplasmic reticulum, vesicles will bud off of that ER, whether it's smooth or rough, and move to what's known as the Golgi complex. It's another membrane system, part of the endomembrane system, if you will. And those vesicles will merge, and the contents of those vesicles will then be modified in some cases. Like, for instance, if we add on carbohydrates or other what we call post-translational modifications. Sometimes we're going to cut those proteins, sometimes we're going to fold them, sometimes we're going to do some of that. That's happening in the Golgi. It's sort of a finalization, if you will. So if we're building the car here in the ER, we're painting the car, installing the carpet and all those things inside the car after the car's been built in the Golgi. The Golgi is also sort of a distribution center. So the Golgi will then repackage whatever it is and send it. It might go somewhere else inside the cell. That's very common. Or it might be going to the cell surface, those vesicles. All right. So the vesicles come in on what we call the cis face. This is the face closest to the nucleus. They get run through here, and you can see this arrow kind of going down step by step. And then on the trans face, the face closest to the plasma membrane, they'll release more vesicles. And again, some of these will go to the cell surface. Some of them will go to other parts of the cell. It depends on what the material is and how it's targeted. And so on to our next organelle, the mitochondria, something that many people are familiar with already. It is the site of what we call citric acid cycle and electron transport of the part of making ATP. So you have two membranes. You have an outer membrane and an inner membrane, and that's super important. You actually use a gradient. We're using a diffusional gradient in between the membranes in order to make the ATP. So it's the site of oxidative ATP synthesis. The prevailing theory about how mitochondria came to be is that what they call the endosymbiont theory, and you don't have to know that, but it's a theory. Both chloroplasts and mitochondria are believed to have arisen this way. That what you had were two separate kinds of cells, and eventually they began working together in a symbiosis way to the point where eventually the mitochondria got enveloped inside the other cell. Mitochondria has its own DNA. You have mitochondrial DNA that you inherit from your mother. It doesn't mean that you don't get a little bit from your dad. There is some swapping going on uh, uh, during development, but the bottom line is that generally speaking, mitochondrial DNA is maternal. So you have just a little bit more DNA from your mom than from your dad. Uh, Depends on how energetic the tissue is and how differentiated the tissue is. Because, like for instance, um, cardiac muscle can't afford to run out of energy. Otherwise, you'd go into cardiac arrest. So you do see abundance in not only in tissue, but where in the cell you find it. Muscles, certainly skeletal muscles, have lots of mitochondria. This fun one here, brown fat. You have two different kinds of fat. You have yellow fat or white fat, and then you have brown fat. And brown fat doesn't get all that much attention, but brown fat is actually a thermogenic tissue. It's not actually for storing energy molecules, it's actually for generating heat. You got a little bit under your shoulder blades, and you have some right under the top of your sternum. It's right up in there, kind of near the thymus gland. It's right in there. Keeps the heart warm, that sort of thing. It's kind of interesting that those thermogenic tissues are near where your heart is, because they are there to do nothing but generate heat. And what they're doing is they are generating ATP through a process called oxidative phosphorylation. All right. We don't, there are some other ways we can make ATP anaerobically, but this is where we make most of the ATP most of the time. And so here's a photo of brown fat. Brown fat is this reddish looking cell right here, and you can see some fat droplets inside it. That's a brown fat cell. And this is these big blobby things are white fat. And white fat has a big lipid organelle inside of it where triglycerides are stored. Triglycerides are what make up fat. Lysosomes, we've already mentioned. Lysosomes, you can kind of consider them the quote unquote stomach of the cell. You phagocytose or endocytose vesicles. These vesicles are then merged with lysosomes, which have very, very low pH. They're very similar to a stomach in many ways, but it's a cellular level. In addition to that low pH, there's actually lots of enzymes in these things that work well at low pH and will break down proteins and carbohydrates and lipids and that sort of a thing. And this may be one of the reasons why, if you have trauma to a cell, you may rupture these things and they begin auto-digesting the cell. But that's what a lysosome is. Lyso, like Lysol. It breaks things down, and a some is an organelle most of the time. Here's the verbiage about lysosomes. Again, contains digestive enzymes. Specialized environment, that means low pH. That must mean separate from the rest of the cell. It allows cells to recycle waste products and so forth. There's something you might have heard. It's called Tay-Sachs disease. Tay-Sachs disease, what happens is you're missing this enzyme, hexosaminidase A. It's an autosomal recessive disorder, and if both parents have a bad one gene, they might be perfectly normal themselves, but the child inherits both copies of the bad gene, and now the child has Tay-Sachs. What happens is you can't break down the phospholipids, and then that leads to either buildup of phospholipids and inflammation, or in the case of nerve cells, they can't function properly because they don't have the right amount of lipids, and they begin atrophy to die back themselves. Cytoskeleton. Now, these are not organelles. You know why? Because they don't have membranes around them. They're called inclusions. All right? And there are three families. You have microfilaments. That's an actin filament. Yes, they are in muscle, but you also find them all around the inside of the plasma membrane. We call it cortical actin. We have intermediate filaments. These are medium-sized filaments made up of a protein called keratin. So if you want to touch an intermediate filament right now, just grab some of your hair. All right, that's primarily keratin. So it's primarily intermediate filaments. Your skin is primarily made up of cells called keratinocytes. All right, rhinoceros horn and your fingernails are also different density packages of keratin, which is what gives us these intermediate filaments. Very strong, very durable, as you can tell with your fingernails and so forth. And then lastly, the largest of the three is called a microtubule. And these guys are like the railroads of cells. So that video I showed you of those green blobs moving around, those were proteins that were moving in a specific direction on microtubules using ATP. So that's those are the three: microfilaments, intermediate filaments, and microtubules. Just a little extra photo for you. This is just, in this case, the nuclear stain is red. So you can actually see nucleoli in here, and that's really cool. Those are not chromosomes. Those are the nucleolus, which is where you manufacture ribosomal RNA and some other things. This teal color is the actin, and you notice that it's a little brighter around the edges. That's because you're seeing more of it. You're looking at it in profile. But this web of actin actually spreads all throughout the inside of the plasma membrane. So I think it's a really cool photo showing you what's called cortical actin. Those are microfilaments. Okay. And so there you go. This here is a really excellent example of a kind of a line drawing that you might see for testing purposes. I want you to notice where the arrows are pointing. Where's number 2 at? Where's number 3 pointing to? Etc., etc. This is very similar to quiz questions or an exam question. The ability to be able to recognize just kind of fundamental structures. Like, for instance, this is Golgi right here. This is endoplasmic reticulum. You can tell this is smooth because you don't see the bumps on it. Whereas over here, number 4, you have bumps on that rough endoplasmic reticulum. Maybe the toughest one in here, I think, this is probably a lysosome. The toughest one are these little dots here. Those are glycogen granules. Okay. So there you have it. We've got the organelles basically down, the primary ones. There are a few extras out there, but these are the ones. And here a little dad humor for you. Dr. Wolf seems to love dad humor. And so biology, the only science where multiplication and division mean the same thing. Because the cells, in order to multiply, have to divide. Very cute. And since we're moving into the cell cycle, we need to talk about two aspects of the cell cycle. First of all, you have the regulatory and side of things. And then you have the actual division itself. We call that mitosis. Turns out mitosis is actually a surprisingly small piece of the cell cycle in terms of time. You have a beginning phase, what's called the G0. Sometimes if cells will stay in G0 for a lifetime. For instance, your neurons, the ones that are mature and differentiated, stay in G0 basically your entire life. What happens is the cell will grow, and if it is stimulated to divide, it'll go past this G0 checkpoint, G1 checkpoint actually, and then it will begin replicating the DNA. That's what S means. It means synthesis. After you're done replicating the DNA, then you have to get ready for division. You have to double check that the DNA has been correctly replicated, pack up that DNA, get it ready, and off we go into mitosis. So these little stop signs are checkpoints to make sure that things don't go wrong. Do things still go wrong? Sure. Yeah, we've got cancer, we've got all kinds of things. But this is the fundamental layout of how a cell, if stimulated to divide, goes through in order to get to that division and then complete that division. So cell division is 3 events, and we're going to start with the DNA synthesis. That's the S phase of cell cycle. This is where you are copying the DNA. This is called replication, where you have special enzymes that get made during G0, G1 that then get turned on during the replication phase, and you're duplicating the DNA. You have the physical division. Okay, there are a couple things we have to do there. First of all, we have to separate the copies of DNA correctly so that each new, what we call a daughter cell, receives not only all of the appropriate genes, but also an equal number of genes. If you have, like for instance, if you've ever worked with somebody with Down syndrome, what's happened with Down syndrome is that one of the reproductive cells didn't divide properly. And so there are extra chromosomes. And that's the thing about human development. If you have too many, or even worse, too few chromosomes, it can lead to all different kinds of effects. You have a spectrum of Down syndrome. Another disease called Fragile X or Philadelphia chromosome. There's a bunch of them out there, and I don't want to scare you too much. Yeah, the process is usually very, very robust, but errors do happen. You want to get when we have mitosis, first we have to get the DNA organized properly, and then we have to undergo what's called cytokinesis. We have to actually physically separate the cells. And so that's step 3. It's the actual bifurcation of the cell into 2 new daughter cells, and hopefully with the appropriate amount of genetic material. There is a second kind of division out there that we see, and it's called meiosis. This is for reproductive cells, the gametes, sperm cells or egg cells. It would not surprise me if this figure strikes fear in your heart, and I don't want it to do that. What is the big picture in this image? And it's got a lot going on in cell biology. We might spend a few days just on this mechanism alone, but we're not going to do that here. All right, we're not going to do that here. One of the ways that DNA is so awesome is that it serves as a template. Okay, there are 2 strands and they complement each other. All right, they complement each other. So this strand has a complementary sequence to this one, and that's why they can bind together. They have the appropriate hydrogen bonds. When we are replicating DNA, what happens is we split the helix. It's called melting, believe it or not. And in that process, the old strand, which is out here, the original strand, one strand serves as a template for the new strand. That's the complementary strand. Same thing on the other side. So if you were to label this old strand with a little bit of radioactivity, what you would see is that the radioactivity would show up in both of the new strands that are made, because some radioactivity was on one strand and the other, and some radioactivity was from the other old strand. So that's what makes this such an amazing, and the word is elegant, mechanism for correctly replicating DNA, billions and billions of base pairs, practically without error. There's always going to be one rare one out there, but billions, 1 in a billion kind of thing. When we get to that mitosis phase, the easiest way to remember the steps, and you should be able to sort of visually recognize these things. Okay, I know that we got a lot to learn, recognize, identify bones and muscles and all kinds of stuff, but this one's not too bad. During it's PMAT, prophase, metaphase, anaphase, telophase. Prophase, what we're doing is we're packing the suitcases. Prophase is where you see the chromosomes beginning to appear. We were packing those suitcases. They're very, very dense. And the nucleus breaks down. You lose that nucleus. It's broken down. You set aside that material for when you are reassembling the cells after telophase. Then you begin organizing these chromosomes to the point where they line up in this beautiful metaphase plate. You can see it right here. This metaphase plate, what's happening is that one chromatid, you see with the bottom of one right here, and the other are getting pulled in opposite directions. So we line them up right on the equator of the cell, all right? And then those chromosomes split. So you have chromatids here, but when they split, now they're their own chromosomes. They're just one copy of the genome, not two. And so these chromosomes will split, and during anaphase, they are being pulled to opposite ends of the cell. And finally, in telophase, this is the physical pinching. Actin, you remember actin, the microfilaments form a ring around the middle here, and the cell uses proteins to literally physically constrict this area and pinch them into two cells. So a prophase, where we're packing the bags, getting rid of the nucleus, starting to organize the chromosomes. Metaphase is where we get them on the equator of the cell. Then we split those the chromatids, and now once they're split, now they're their own chromosome. So we split those chromatids, we move them to the poles, and then we pinch off the middle in telophase. You can see here now the nuclei have reassembled. So you have nuclear membrane. The chromosomes are no longer immediately obvious, and we're just at the very end of pinching off those cells so that you now have two absolutely complete cells. They should be, on average, an identical copy. Certainly genetically speaking, that should be true. Not always true about cytosol, but we're not worried about that. If you've got a cut and your skin cells are trying to heal that cut, those skin cells should be about as close to being identical as possible. And now on to meiosis. And again, we're going to try to keep this relatively superficial. The funky thing about meiosis is that you have two mitotic divisions. So it's PMAT times 2. You have two independent cell divisions. Okay, two independent cell divisions, which means that you are going to end up with 4 reproductive cells, 4 gametes. Okay, if you divide the cell one time, you've copied the DNA. Okay, so now you've got basically 2 cells, and those 2 divide, and so that gets you 4. When those 2 divide, they don't divide the chromosomes the same way that mitosis does. All right, so be careful here. Just remember that any cell that undergoes meiosis, it goes through 2 mitotic divisions and ends up with 4 what are called gametes, because they have half the number of chromosomes that a normal cell. A normal cell is what we call 2N. It's diploid. These are haploid. They only have half of all of the genetic material that a normal adult cell would have. We see these in the gonads. Ovaries and testes is where this occurs. Okay, ovaries and testes. It does keep from inflation. If we didn't undergo this sort of specialized, first of all, there are divisions. There's also a swapping of genes. It's really amazing. All right. This way, by reducing it, like for instance, let's say you had a diploid sperm and a diploid egg. Then what would happen is when those two merge, that would be 4N, and you'd have to have some kind of special division. In theory, it's possible, but the way that nature has solved this is to do the divisions ahead of time rather than after the fact. All right. So rather than having to split weirdly at the beginning here, the two haploid form an intact cell, a 2N, 46 chromosomes, and then they can just divide like normal after that. All right. I had a genetics professor one time. He just walked into class. Here are a bunch of undergraduate college students that are just full of reproductive energy. You know what I'm talking about, right? Okay. And he goes, well, what's the purpose of sex? And then he goes aside, goes, other than it's fun. Okay. All right. And that's a way to open a class. Okay. Here's the reason. Genetic variability. Okay. If populations reproduce asexually, that means there's very difficult to introduce new combinations of genes because you're just doing the same Xerox copy. Now, that may work in some populations, but there are some other mechanisms involved that involve mutation. And that's kind of weird and that's kind of difficult, especially in a complex organism. So complex eukaryotic organisms generally go through sexual reproduction because it allows for what we call recombination. So when we're making these meiosis divisions, there's a step where chromosomes from one parent and another parent will actually come together and swap material. It's really amazing. All right. It's an actual structure. They've described it. And so you swap genes. And maybe you get a few genes from your dad, but then you get a bunch of genes from your mom on one chromosome, and then maybe the other way around on another chromosome. That recombination, that variability means that the offspring may see a new combination that's more favorable, or in some cases less favorable. That's that's just the way it works. It allows for combination and what we call independent assortment. That is, the genes get randomly reassorted when you create these gametes, and that's why we say biodiversity. You hear that phrase a lot. More diverse populations are stronger. They are more able to withstand potential ending events, shall we say. One of the reasons why cheetahs in Africa are so vulnerable, is because they are everything but identical twins. And so all of their recombination and independent assortment is basically the same. Because what they think happened was that there was such a huge collapse in population that you were down to just a few individuals. And so you basically ended up with they're like 93% identical. So if identical twins are basically 100% or pretty close to it, these guys are in the low 90s. There's not a lot of variability there, which means that if something bad were to happen, a disease were to come through or something else, it could just wipe them out. Okay. So I've talked a lot about there. I didn't mean to go that long, but I thought you'd find that interesting. I hope you did. And so that finishes part 2, cell division and the different organelles that we talked about. Again, try to get that big picture first and then fill in the details. Rather than trying to learn all this information linearly, start with the big picture, the message, and then get a little more detail next time and try to add a little bit to understand division, right? Okay. All right. Well, good luck. Thanks for your time. I hope this was helpful. And I will see you in class real soon. Take care.